Okay, so we'll tell about mitosis on the basis of the mitosis 3D animation phases of the mitosis cell division by the creative learning. As usually, I will stop from time to time.
In this module, you will learn about mitosis. Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into two identical daughter nuclei. It is divided into four key phases: prophase, metaphase, interphase, and telophase. An additional phase called cytokinesis occurs after the complete nuclear division.
So cytokinesis is when we have already two cell nucleuses, so everything complicated is finished, and we need just to divide the rest of the cell. Yes, so as I pointed out last time, the complex part, the difficult part, is to create new cell nucleus. Dividing rest is relatively simple.
Dividing cell enters mitosis. It undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases. Prophase.
So just not to mess. Interphase is not mitosis. Interphase is between mitosis. You will answer me more about the interphase in the next question.
During prophase, the chromatin condenses and becomes short and thick to form chromosomes.
Yeah. So this is important thing to remember, yes? A lot of you think generally that we have the chromosomes all the time in the cells. And it's not true. We have the chromatin most of the time. The chromosomes we have only, they are appearing only at the very beginning of the cell cycle, of the mitosis, and meiosis also. In the prophase, they are forming in the prophase. That are not the biggest, they are the best seen during the metaphase, generally. They're the biggest during the metaphase, but they are forming during the prophase.
Each chromosome has two sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down.
So those chromosomes are like free-floating.
The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface.
So kinetochores are generally attached to the centers of the chromosomes.
The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores and the daughter chromosome.
Yeah, so during the anaphase, those chromosomes break up, just to give two identical pairs. Nearly identical. Yes, of course, not exactly.
Along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into two sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane.
So generally during the telophase, we are recreating the normal cell nucleuses. But now we have two.
Cytokinesis.
And what is important, we do not have reduced amount of the DNA because the cell had prepared for it. Because before, during the interphase, you already read more about this, during the interphase, the cell created additional amount of DNA. So now, when it would divide, it would not lose the DNA. It would just lose this additional part that was created, that was prepared for the mitosis. Okay, thank you very much.